Hey guys, so this is meiosis. And meiosis is also known as spermatogenesis, which is the creation of sperm. And later we'll talk about oogenesis, which is the creation of egg, okay? And meiosis occurs in gamete cells, or also known as sex cells. And the goal of meiosis is to cut the number of chromosomes in half, okay? Why would this occur? Because as we know that all gamete cells, like, they're haploids, right? But it's like they all start off from a diploid cell. So why do we want to cut this in half? Well, because during fertilization, when the sperm fertilizes the egg, if the sperm has, for example, in humans, if the sperm has 46 chromosomes and the egg has 46 chromosomes, when they combine, then the offspring would have 92 chromosomes, which is not what we want. We want it to stay 46 chromosomes. So the goal of meiosis is to make those 46 chromosomes into 23 chromosomes, at least in humans, and the number varies on different species. Okay, sorry. So in meiosis, you start with a diploid and you end with haploids. I didn't draw here, but in meiosis, just same like we see in mitosis, cell goes under G1, S, and G2 phases. Same thing. S phase, they duplicate their DNA. All the gist. Okay. So in prophase 1, which is the first step of the meiosis stage, we see here. So a few things happen. Okay. So first we see the dissolving of the nuclear membrane. We see how our duplicated centrioles start to move towards the poles. But more interestingly, we saw that our homologous chromosomes had condensed and created sister chromatids. So these, we have two sister chromatids, like two X's, but these are homologous to each other. Okay, the reason why they're homologous to each other is because before this prophase 1 step, we had a cell, and then we had, for example, one blue and then one pink, right? And these are homologous to each other, okay? But then the pink one duplicated itself and the blue one duplicated itself, okay? There's still, and then the blue crossed over, the other blue, and then this one crossed over, and then that crossed over that, right? So we got two X's. But the thing is, these X's, these sister chromatids, are still homologous. The chromosomes are still homologous chromosomes, but they're just consist of sister chromatids now. So we see here something that happens in prophase 1, and specifically unique to prophase 1, is that synapsis occurs. Synapsis is where our homologous chromosomes are one X and the other X. They do something called synapsis, where they snap together and they form a structure called a tetrad. Okay, tetra, in like math or anything, it means four. So that's why, because it's a tetrad, because it's four sister chromatids. It's so the homologous chromosomes, they snap together in synapsis. What we also see in prophase 1 is that crossing over occurs. Crossing over is where one section of one sister chromatid will trade with another section from there. So they're kind of swapping traits, okay? But it's like still the same traits. Like, for example, say this includes the hair genes. This also includes the hair genes. They're just kind of switching it. And this is actually the first reason why siblings are different, because if you and your sister both come from the same parents, how are you guys not clones of each other? There are three reasons. First reason is that the crossing over of traits is completely random. It's completely random. And it may not occur in all cells. And I'm sorry, it may not occur in all homologous chromosome sister chromatids, but it does occur in most of them. So from prophase 1, you go to metaphase 2, okay? Remember PMAT, so then you go to metaphase 2. What occurs in metaphase 2? So unlike metaphase, just normal metaphase that we had in mitosis, the homologous chromosomes don't line up at the equator in like a straight line. Actually, the second reason why siblings are different, the second reason why you and your sister, your brother look different is because the deciding of the lining of the homologous chromosomes is completely random, which meaning like which one is gonna go up and which one's gonna go down. It's completely random. There's no like really like a depending factor. Like you can see here that this one was kind of at the top, but then this one ended up at the bottom. It's completely random. That's why you and your siblings. Second reason why you and your siblings look different. So the first reason why you and your sibling looks different because of crossing over, which is completely random, and now the lining of the homologous chromosomes during metaphase 1 is also completely random. And then in metaphase 1, we see how the centrioles have radiated spindle fibers, and then the spindle fibers attach to the centromeres of our homologous chromosomes. We know this. Third step, anaphase 1. So then the spindle fibers that attach the sister chromatids start to shorten. And now because these are not next to each other, these are not considered homologous anymore. They're just known as sister chromatids, okay? And then the continuous spindle fibers that go from centriole to centriole are called, they get longer, and then that causes they start to get longer in anaphase 1. And then in telophase 1, it's kind of like oriented differently, but then the whole cell has elongated, and then cleavage furrow occurs. And basically you saw here how cleavage furrow occurs, right? So it like kind of cuts off this part from that part, and that's why you see how inside each one of these cells it's two X's. And also the sister chromatids, they uncondense and they make chromatin, and they're not homologous anymore. And then cytokinesis occurs, pinching is complete, and 2 full cells are created. But these 2 cells are considered haploids. They're not considered diploids. Even though you see like, oh my god, there's two, how is it not a diploid? But the thing is, the definition of a diploid is the fact that the pair is the homologous chromosomes, both chromosomes are there. But remember that this one and this one and this one were separated. These were once homologous, but then they were separated. So this is why the product of cytokinesis 1 is the formation of 2 haploid cells, okay? But this didn't cut the number of chromosomes down, okay? So how are we going to cut the number of chromosomes down? Well, after cytokinesis 1, instead of entering the G1 phase and all of interphase, and especially S phase, where it duplicates the number of chromosomes, it skips that. It might go to G2 to duplicate the organelles, but the main thing is that it skips S phase so that you're not duplicating the number of chromosomes, because our whole goal is to cut the number of chromosomes in half, okay? And then from cytokinesis 1, it goes to prophase 2, okay? And again, we see the centrioles moving. The centrioles moving to the poles, okay? And then we saw how these sister chromatids, the X's, they actually condensed again. So now these are sister chromatids. And then the nuclear membrane also dissolves. And then the next step, and the difference between prophase 2 and prophase 1 is the fact that in prophase 1, crossing over occurs, but in prophase 2, crossing over does not occur. Metaphase 2. So then, unlike metaphase 1, these sister chromatids, they line up at the equator, just like how we saw in the normal metaphase in mitosis. They line up at the equator, spindle fibers attached to the centromeres, and then spindle fibers attached from central to centriole. We know this. We've done this a lot of times, okay? And then in anaphase 2 is where now the spindle fibers are pulling the sister chromatids apart. So now we don't have sister chromatids anymore. We just have individual chromatids, okay? And this is occurring in 2 cells, okay? It's not just occurring in 1 cell. It's occurring in 2 cells. And then the continuous spindle fibers, they elongate as well. So that causes the telophase for it to stretch. And then we see cleavage furrow occur in both. And then we also see how these chromatid uncondensed into chromatin. And then cleavage furrow occurred, nuclear envelope is back, and we have telophase 2. And then the last step is cytokinesis 2, where now the 2 cells, they fully separate into 4 separate cells. And if you can see, these 4 cells, they have exactly, each one has exactly half the number of chromosomes as the beginning parent cell. And what else? So yeah, these are, as you can see, like, remember, because these are haploids, they are not diploids. Even though you see, because you don't see like 2 long ones and 2 short ones, you see a long and a short one, which means that the long pair is not there anymore. So that is cytokinesis 2. So you start off with 1 parent cell with, like, for example, 46 chromosomes, but then you ended up with 4, and this is happening in male, so you started off with 1 male gamete parent cell, and then you ended up with 4 sperm cells, each with, for humans, it would be each with 23 chromosomes. And the third reason why you and your sibling are different is the fact that you have 4 chromosomes, but only 1 egg created, because oogenesis only creates 1 egg, and I'll talk about that. So that's the reason why, because depending on which sperm fertilizes the egg, that carries different traits, and that's why you and your siblings look similar, because, you know, you have the same parents, but also you have unique traits because there were 4 different sperm for 1 egg. Anyways, so what is oogenesis? So oogenesis is the production of egg. And oogenesis, unlike spermatogenesis, where 4 sperm are created, oogenesis, only 1 sperm is created. How? So basically, we start off with a parent cell at the top, okay? And then, you know, all of meiosis 1 occurs. The steps are pretty much the same. So all of meiosis 1 occurs, right? So we get, but the thing is, what happens is that after cytokinesis 1, okay, after cytokinesis 1 egg gets more cytoplasm than the other egg, okay? So that causes 1 egg to be bigger than the other egg, okay? And that was only meiosis 1. So then these 2 are going to go under cytokinesis 2, and this is going to split in half again. And again, the same thing is going to happen where this egg, it's going to give most of its cytoplasm to this egg, and this is going to turn out as the thriving egg. And then this one is going to get like less cytoplasm. So then these like small ones, they are called polar bodies, and they don't really help humans, but they are useful in plants, and they just eventually disintegrate, okay? So that's how you created 1 egg. Yeah. Thanks so much for watching. Bye.